Tonight's program is sponsored by the College Scholars Program. This program provides a stipend to JCCC faculty, both full and part-time, for the development of 2 research presentations, one of which, of course, is taking place tonight, and either a guest lecture in the classroom or a faculty seminar on the topic of the presenter's choice. College Scholars was started by Dr. Jim Leiker to provide a platform for JCCC faculty to present their research projects. Those of us who are involved in this program feel it's important for a number of reasons. First, community college professors, of course, the emphasis is on teaching. So this gives community college faculty a chance to explore their own research interests. Second, I think that pursuing your own research interests makes faculty, here we have 2 extraordinarily wonderful examples, better teachers. Because they can bring new insights and new enthusiasm to the classroom. And I think there's a lot of students who are students of these professors who would say that's exactly what happens in their classroom. And then finally, it's a way for the community to understand the resource that they have at JCCC and the resource that they have in the faculty. So community members have come to every one of these presentations, and there's always a lively back and forth and exchange of questions and answers at the end after the presentation is over. So it's a chance for the members of the community and faculty to meet and get to know a little more about each other. So now on to tonight's speakers. Heather Seitz received a PhD in microbiology and immunology from the University of North Carolina, Chapel Hill in 2006 and joined the faculty at JCCC in 2007. Melanie Harvey received a PhD in inorganic chemistry from Vanderbilt University in 2000 and joined the JCCC faculty in 2008. Both Dr. Harvey and Dr. Seitz have published extensively in scholarly journals and have presented also extensively at academic conferences. They are also sought-after speakers for both their teaching and research expertise. As professors at Johnson County Community College, they have collaborated to turn their classrooms into living laboratories and encourage their students to become part of the solution to the problem of antibiotics resistance. Dr. Seitz's students run experiments on local soil samples to isolate bacteria, while Dr. Harvey's students characterize the antibiotic compounds. Dr. Seitz and Dr. Harvey are 2 of about 200 educators around the world who are engaged in this type of experimentation in their classrooms. They are part of the Small World Initiative, a program started at Yale University that has the dual purpose of encouraging students to pursue careers in science and to crowdsource the search for new antibiotics. Their innovative teaching and their commitment to JCCC has won Dr. Harvey and Dr. Seitz numerous awards, among them the Distinguished Service Award and the Burlington Northern Santa Fe Railway Award, both of which are presented to faculty who demonstrate excellence in teaching and service. I am very pleased to have both of them here tonight. Ladies and gentlemen, Dr. Melanie Harvey and Dr. Heather Seitz.
Good evening, and thank you, Sarah. That was a very warm introduction. So tonight's talk is going to be about the challenge that we're facing with antibiotic resistance. We're going to be talking a little bit about the statistics on antibiotic resistance in the United States today, the history of antibiotics, how they work, and how is it that bacteria can become resistant, some important classes of antibiotics, and we'll talk about the antibiotics of last resort, as well as discuss a little bit of the chemical structure of those important classes. We'll be talking about what's being done to develop new antibiotics, both here at JCC, as well as nationwide and international efforts. And lastly, we'll talk a little bit about our research into finding new antibiotics. That will be primarily the focus of our second talk on Wednesday, April 19th at noon in this exact auditorium. So if you'd like to come back to hear more about our specific research results, feel free. We would love to have you. So we take antibiotics for granted. Today, my son is sick and probably has strep throat. I know that tomorrow I will probably take him to the doctor and he will be prescribed an antibiotic and he will get better. I have faith in that, right? I know that that's the reality that I live in right now. But that reality is precarious, right? I want to introduce you to a little girl. She's 2 years old. She is from Connecticut. And in September of 2016, she went on vacation with her family in the Caribbean. And it was a wonderful 2-week vacation filled with lots of food from the local markets, including goats and chicken meat. And they had a wonderful vacation. But about 2 days before they returned to the United States, this little girl fell ill with some diarrhea. Immediately upon return to the United States, they took her to the pediatrician, right? And the pediatrician was involved in a new effort called the Pathogen Detection Network. So they took a sample from this little girl's stool from her diarrhea and sent it to the Pathogen Detection Network. They immediately began treating her with an antibiotic. Because she had been visiting the Caribbean, and there's a lot more diversity in the types of pathogens that you might see in the water and the food samples there, they treated her with a slightly unusual antibiotic for that case, parmamycin, which is primarily prescribed for amoebic dysentery, or a parasite-like dysentery that we typically don't see in the United States. That turned out to be one of the best things that they did for this little girl. She recovered just fine, and she was released from the hospital and made a full recovery. But that entry into the pathogen detection network showed us how close a call she came, right? So they analyzed the sample by DNA analysis and found an important antibiotic resistance gene called MCR1. And it turns out that the bacteria she was infected with, E. coli, had this resistance gene. This is an antibiotic of last resort. So had they used the traditional antibiotics to treat her, they would have found that no antibiotics that they had tried would be effective. That is not an isolated case, unfortunately. Each year in the United States, more than 2 million people will get sick every year here from an antibiotic-resistant infection. That is more than the entire state of Nebraska, which is where I'm from. Right? So I care about those people. And of those 2 million people, 23,000 of them will die every year in the United States. That is half the student population at Johnson County Community College and the entire population of K-State University. So that's a lot of deaths from antibiotic-resistant infections. So what happened before antibiotics?
Okay, so before antibiotics, your chances of dying from a bacterial infection were very high. In fact, in 1910, 46% of the deaths in the United States were from infectious disease. By the year 2010, that number had dropped to just 3%. The difference is both vaccinations and antibiotics. Now, people were using antibiotics before they realized they were using antibiotics. We do know that some ancient cultures used moldy bread in infected wounds or in wounds to prevent infection. And so they didn't even realize what they were doing, but they were, in fact, using an antibiotic. We've also found evidence in mummies. They found evidence of tetracycline. And they have deduced that it was in their process of brewing beer that they were actually also making, growing a bacteria that produced tetracycline. So in their regular diet, they were taking antibiotics. And they've also seen that Egyptians that were in this group had a lower incidence of bacterial infections. Okay, so the miracle drug that I want to just tell you about the story of a little bit is the first time that antibiotics were available worldwide, and that is penicillin. This is Alexander Fleming, and he is credited with as the first person to publish the discovery of penicillin. And he was studying bacteria. He went on holiday for 2 weeks, and he came back, and here is a photograph he took of what he found, and this is a plate where he was growing. These little white dots represent, these actually are staph. So he was growing staph, but then there was this contaminant, and it turns out that contaminant was Penicillium notatum. And what was interesting about this that stood out to him was the fact that in this whole region here, you see that the staph, it looks like the staph was there, but it died. Okay, so then he started studying this Penicillium notatum to see what is it making, what is it producing that is killing the staph. And he ended up, at first he called it the mold broth, but then he shortened it to just call it penicillin. So he would extract the penicillin. He did have a little trouble isolating the penicillin, specifically the chemical, because it was very unstable, the way he was trying to isolate it. So he would just use this mold broth. So then some folks at Oxford worked on this. Norman Heatley first was able to successfully extract the penicillin drug molecule, and Edward Abraham used a new technique at the time, column chromatography, to help remove impurities and purify it. And then Howard Florey, in 1940, he started testing it on mice that had bacterial infections to see, if it would cure their bacterial infections. This process required, at this time, a lot of this mold broth. And so they hired a bunch of ladies, they called them penicillin girls, and they paid them hourly to maintain these cultures and to grow lots and lots of mold. And in fact, they needed, at that time, to do the testing that he was doing, they needed 500 liters a week. And the reason I spelled mold like this is really because these folks are all British, and so I just went with their spelling, right? Okay, so then Florey used it on the first patient, and I believe I read that it was his wife who knew of somebody, and they just decided to try it out on this guy. So there's this policeman, and he is 43 years old, and he was pruning his roses, and he accidentally scratched the side of his mouth, but then he developed a horrible infection. In fact, it was so bad that it had taken over, he had abscesses on his face, and they had to remove one of his eyes, and it was infecting his lungs, and so he was going to die. And they thought, well, this is the kind of candidate that we want to try this on, is somebody who, if he was going to die if we don't do something. So this is where they started. They injected him with penicillin, and he made a remarkable recovery. He was eating, feeling better, but then they ran out. Because remember, they had all these 500 liters a week, which just to test some mice. So they needed a lot more for a human. And unfortunately, they ran out of it, and he died 5 days later. So at that point, they said, okay, we're only going to use this for our first clinical trials. We're only going to use this on children at this point, and only when they're as a last resort, to try to save them. And that way we won't need as much on a child as we would a grown man. So this is the first, here's an example of first girl. I believe that she was the first one, right? Yeah. And this was in 1942. And here is, you can just tell from this picture that she is in a really bad state with an infection, a bacterial infection. Picture 3 is after 4 days of penicillin. Picture 4 is after 9 days, and then 5 and 6 shows after she's made a full recovery. So this was kind of amazing, because if you imagine how many people were dying, and just imagine, cutting yourself, pruning roses, and that's a death sentence. That seems bizarre to us today. But this is really the reality of what people were living in when they didn't have antibiotics available. Okay, so they needed a lot more. And it was in the middle of World War II. And, Britain was in no shape to be mass producing something like this. All of their chemical plants were focused on the war efforts, and they did not have, it was not a good time for them to be mass producing penicillin and to sort this out. So they looked to the United States for help. And this guy is Andrew Moyer. He's the director of the USDA's Northern Research Laboratory in Peoria, Illinois. And he was doing agriculture research. And one of the things he was doing was he had a lot of byproducts from the wet mill processing of corn. And he's always trying to find a use for that. So that was just a thing that they seemed to try it on everything. But he worked out that if he added this corn steep liquor to the fermentation medium that he was growing this mold in, and he substituted lactose, which is the sugar in milk, for sucrose, which is table sugar. He substituted those in the culture medium that he's growing it in, and he actually increased the yield tenfold. So suddenly now they can grow it 10 times as much penicillin in the same volume. And so then this became like an important part of the war effort, really. If you imagine all the soldiers that are being injured, and a lot of their injuries are susceptible to infection, this was very important that they really mass produce this and make it available. So basically the War Production Board was formed, and they talked to all the chemical companies in the US, and they chose 21 companies to participate in this effort. And they would grow these molds in big fermentation vats where it was aerated, and they really maximized how much they could get. This was a big production. And Pfizer was the first commercial plant that was opened. And I just wanted to show you this because I think what's interesting, and it started making me think about the World War II generation even, and that sense of, at that time, of not so much individualism as much as like doing things for the good of everybody. They really had very much of that sense of sacrificing for the many. But they did this whole campaign because they needed people to get on board. It took a collaborative effort of more than one country, and it took scientists that were working in government, in academia, in hospitals, in in chemical plants, in industry. It took all of them working together to bring about this mass production of penicillin and make it available to the world. So I think so they launched this whole campaign of just letting people know, like, this is important to everybody, and we should care about this.
All right, so we've learned that penicillin was originally made by a fungus. Right? So we know that fungus can make antibiotics, but bacteria can also make antibiotics. And you think that's interesting because these drugs are designed to kill bacteria and to kill fungi. But bacteria do this, they make these antibiotics as natural byproducts in their environment. So we all know animals of every species, and bacteria and fungi included, they all want to stake out their own little claim, right? Their own little picket fences, as you were. So they all want to compete for the space and nutrients in their local environment. And an environment like the soil has millions of bacteria per gram. So they all want a little piece of that. So these bacteria and fungi make these compounds, these antibiotics, to help secure their little space, to make sure everybody else stays out of their little zone. And I'll show you a picture of what that looks like on an agar plate later in our talk. So think of it like little picket fences that these bacteria are producing. Now, it's really important that the compounds that a bacteria makes don't kill that particular bacteria. So I want to take you all back to biology class for a minute and talk about the difference between a bacterial cell and a human cell. So bacteria over here on the left are much smaller than their human cell counterparts, and they are much simpler in design. They have very few components to the cell. And there is a wide variety and diversity in how these bacteria are created. So we have bacteria with a certain type of yellow coating or cell wall called gram-negative bacteria. And we have other bacteria that have a very complicated, thick cell wall that are called gram-positive bacteria. So there's lots of different kinds of bacteria that have different components that are shown here in this cartoon diagram. And that's important because as bacteria create an antibiotic, that antibiotic is oftentimes for a different kind of bacteria than they are. So gram-negative bacteria might make an antibiotic that harms gram-positive bacteria, for example. Now, these cells are very different than human cells. So if we are going to take an antibiotic, the goal of that drug is to kill the bacteria without harming our own cells. So these drugs also have to be designed to not harm any of the complicated workings of our own cells. So hopefully you can see from this cartoon that there's a lot of stuff going on inside that cell, right? There's a lot of organelles and complicated machinery that all works together inside of our own cells, and the drugs that we take can't harm any of that. So it becomes very difficult to pinpoint those exact differences that bacteria have that human cells do not have. So how do these antibiotics actually work? So antibiotic drugs target many different places in the bacterial cell that are different and unique from those human cells. So we know that there are antibiotics that target the DNA structure. So this is the instructions to make all the proteins of the cell. So your DNA encodes all the things that you need, and the enzymes that copy the DNA and RNA in a bacterial cell are slightly different than the ones in human cells. So we have a whole class of drug that targets those enzymes. We have antibiotics that target the cell wall. We'll talk more about penicillin. You've already heard a little bit about penicillin, but we'll talk more about how it works And those are drugs that target that yellow coating around the bacteria that is definitely different than what we have in our human cells. We have drugs that target the cell membrane and drugs that target the ribosomes. So these are the little machines inside of your cell that take your DNA and turn it into proteins. Right? And there's slight differences in how these ribosomes are made to how human ribosomes are made. So there's whole classes of drugs, and you can see the list of the different antibiotics here, that target those ribosomes. And lastly, it's important to note that there's a class of drugs that work to inhibit the metabolism of a bacteria. So bacteria can live in places that we can't live, right? They can live in toxic waste, they can live in the soil, they can live without oxygen. They can do amazing things that we cannot do. And most of that is because of enzymes that are involved in their metabolism and growth. So we can use antibiotics to target those differences without harming our own cells. So how is it that these bacteria become resistant to antibiotics? So every time we try to pull out a weapon, they pull out a bigger sort of defense. So bacteria are smart and they're very adaptable. Their DNA can change rapidly, much more rapidly than human DNA changes over time. And those changes in the DNA can lead to some important resistance factors. So you can have a change in the bacteria's DNA that does not allow the drug to get into that cell. So a change in the DNA changes what this cell wall looks like, and the drug can no longer get in. You can have a change in the DNA of the bacteria, and now an enzyme that may have been involved in metabolism can now break apart the drug, the antibiotic drug, so it can just chop it in half. You can have a change in the DNA that alters the place where the antibiotic binds. So antibiotics are these little red balls, right? So if the antibiotic is supposed to bind this component, a change in the DNA can change that protein to a different shape, and the antibiotic can no longer bind. And the last mechanism that I want to talk about, and one that you'll see sort of us talk about throughout the talk, is you can change a pump. So this is a channel through the membrane, and bacteria have lots of these for transporting things across the membrane. And you can have a change in the DNA that alters this pump, and so instead of pumping out the metabolism product that it was supposed to pump out, it now pumps out the antibiotic, right? Think about when you try to feed a baby peas for the first time, they just spit it right back at you, right? That's what I see in my head, right? So that efflux pump, it just pumps it right back out. And that's a really effective way for these bacteria to become resistant. So how do these changes in the DNA happen? So changes in the DNA happen, they can happen randomly. So every time the bacteria divides, it copies all of its bases. It's millions of bases of DNA. And sometimes the enzymes that do that make a mistake. So you can have a random mutation. Those random mutations are more likely to occur if the environment that the cell is in is stressed, meaning it's not quite the right temperature or not quite the right oxygen level, or it contains antibiotics. So if you have a suboptimal level of antibiotic drugs, so a low level of antibiotic drugs, it's not quite killing the bacteria, but it's causing a lot of stress on that bacteria. The bacteria is having a hard time doing its job of metabolizing and growing. You are more likely to have these mistakes or mutations occur. So here we see a change in the DNA that's caused randomly. And it's created an efflux pump here in this little red arrow. In a population of cells where there is one mutant bacteria, this mutation can slowly change over time, right? And I want to show you an example of sort of in real time how these random mutations can occur. Now, before we look at this video, I want you to know this is a high-stress environment. So one of those environments where you see lots more mutations than you might otherwise see. Okay. So again, that's unlike what you would see in the human body. We don't have, thankfully, increasing amounts of antibiotics over time, but with that selective pressure, with that increased amount of antibiotics, you can accumulate those mutations at a pretty rapid pace. Now, that is not the most dangerous type of mutation or antibiotic resistance that we're worried about. Bacteria can share their DNA. I love this cartoon. It was on a shortcut through the hospital kitchens that Albert was first approached by a member of the antibiotic resistance. So we see two bacteria here, and this particular bacteria is looking a little shady, and he's sharing his DNA with another bacteria. And this is a mechanism that bacteria can do. They can use their DNA can be on mobile elements called plasmids, and these are specially designed pieces of DNA. They are designed for sharing. So bacteria in their natural environment, they do clonal proliferation, meaning they clone and copy themselves identically. It's not a very good idea for robust diversity. So humans, we don't clone each other, right? Because we like the diversity that we see around us, and that makes us a healthier population. So bacteria have these mobile elements to help them survive in complex environments where there might be additional stress. And so bacteria of the same species, right? These guys look sort of the same, can share this bacteria on plasmids. But interestingly and scarily, right, there are bacteria of different types can share their DNA. So in our opening case with the 2-year-old from Connecticut, she actually was infected with a strain of E. coli that had at one point gotten some DNA from a Shigella bacteria. So different kinds of bacteria can share their DNA and cause resistance. And not just different bacteria, but viruses can infect bacteria and share DNA and increase the spread of these mobile elements. Now let's think about that for a second as we look back at our example of a population. So if a bacteria is introduced that has this mobile element, so notice the red mutation here is on a separate piece of DNA, one that's easily shared between different bacteria, we can much more quickly see the spread of these antibiotic resistance genes. So that gene that that little girl had from Connecticut was actually on one of these mobile elements, which makes it an incredibly scary type of infection. Now, I want to talk for a second about the diversity that we see in bacteria. So we've talked a little bit about how bacteria can come in different shapes and sizes, but in our gastrointestinal tract, imagine that we have a collection of bacteria. Most of that bacteria is good bacteria. It does good things for us. It helps break down our nutrients. It helps us digest our food. It helps us stay healthy because there's not space and nutrients for the dangerous pathogens to set up shop. There's a lot of research now talking about the human microbiome, right? So you can send off and find out what your microbiome profile is. So we know that this is a very complex and diverse environment. If we have this mutant bacteria and we destroy all of the competition in that environment, the only thing left is this resistant bacteria. So how would we go about doing that? Well, when I take my son to the doctor tomorrow, he's most likely going to be prescribed a broad spectrum antibiotic. And what does that word mean? Well, broad spectrum means an antibiotic that is effective against many different types of bacteria. In many cases, it kills off a lot of the normal bacteria that you have in your gastrointestinal tract. Remember, these drugs don't know which one's the pathogen and which one are the good bacteria. So in the case of a broad spectrum antibiotic, if we have this one mutant bacteria, we've killed off all of the competition. We now have more space and nutrients, right? What we see is a selection for that resistant bacteria. So every time we use antibiotics, we select for those that are resistant. And in the case of a broad spectrum antibiotic, that is all that's left. So what happens if we take a narrow spectrum antibiotic? And these are drugs that are harder to prescribe because you have to be more confident about what kind of infection your patient might be experiencing. So a narrow spectrum antibiotic is only going to work against one type. And so if we do that in this situation, we kill off only one kind of the bacteria, and we're left with those good guys that can then proliferate and create that coating to help us protect against the pathogen and the potentially resistant bacteria. So they can help us compete for space and nutrients, and we see much less spread of that mutation, that resistant bacteria. So how do these antibiotic-resistant bacteria spread in a population? And this graphic is from the CDC, and it starts here with George, right? He got some antibiotics that applied some stress in his gastrointestinal tract, and he developed a resistant bacteria in his gut. So he may have gone and then been a part of a healthcare facility like a hospital or nursing home where the people in those that he encounters in that environment are highly susceptible to these types of infections. So that is one way. We call that sort of hospital-acquired resistance. Or George could have gotten better and gone home, and he could still have passed it around with his friends and family. You share a microbiome with the people that you're closest with, right? So you have similar bacteria. And so George could have shared those resistant bacteria with the people that he cares about. Now that is one side of the story. So when people have those resistant bacteria, they can spread them in the population. And that's the side that we typically think of. But we also have to pay attention to this other side. So in agriculture, there is widespread antibiotic use. And so those antibiotics inside the animal gut also can develop resistance. And if we don't handle or cook those meats properly from those animals, we can also be exposed to those resistant bacteria. And the fertilizer or other water that's been contaminated with those animal feces can also spread those drug-resistant bacteria through our vegetables. Right? So these resistant bacteria can spread in the population in many different ways. Now, what can be done about this? Well, this is the CDC report from 2014 talking specifically to prescribers, so doctors that prescribe antibiotics. And they say order recommended cultures before antibiotics are given. So let's think of the last time a kid that you know or you suffered from bronchitis or an ear infection. Did you walk out of the doctor's office with an antibiotic script? Did you wait 48 hours before getting that antibiotics? No, right? Now, if that's the first recommendation every time we prescribe an antibiotic, why isn't it being followed, right? Well, we don't tend to go to the doctor when we're kind of sick, maybe getting sick, right? We go to the doctor as a last resort. We are very sick. Our child is very sick. We can't miss any more work or school or whatever commitments we have. We need to get better now. So patients have a responsibility in this. We don't want to wait 48 hours. Can you imagine if a kid has a terrible fever for 2 days, you take him to the doctor and the doctor says, yeah, just sit on that for 2 days and I'll call you back, right? As parents and even people that have been sick, that's not acceptable. The other side of this is the money, right? So it costs a lot of money to run the tests to prescribe those special narrow spectrum antibiotics. The insurance companies don't want to pay for that. You don't want to pay for that. And it takes a lot of time and resources. So it's very difficult to follow this first recommendation for physicians. They should also make sure that the dose is appropriate and to make sure to check back in 48 hours later and adjust the prescription, right? Which also doesn't always happen at the rate that we'd like to see. Now, we can't always control what we walk out of the doctor's office with, but some behaviors that you can contribute to that can help us reduce the spread of antibiotic resistance. Don't seek antibiotics for those viral infections. Remember, every time we use an antibiotic, we select for resistance. We increase the number of antibiotic-resistant bacteria in the environment. Don't take antibiotics that are just lying around, right? So if somebody didn't finish their antibiotics, we'll get to that one in a second, but those antibiotics are either expired, you might be allergic to them, they are prescribed at a dose that is not you, like so if you take little kids, that is not the same size as me, right? Or they are old and expired, so there's lots of reasons why you shouldn't take antibiotics that you have in the cabinet. Don't stop taking your antibiotics until they are gone. So remember that the stress that we put these bacteria under when we take antibiotics can increase the rate of their mutations. So if you take antibiotics just long enough to feel better, but you haven't killed all those bacteria, they will remember. Right? They are more likely to have those mutations. So you want to make sure that all of the bacteria that you've been sick with are dead, right? They are gone. So take that entire prescription. And should you have any issues with antibiotic resistance, you always want to make sure to talk to your doctor promptly if you don't feel better after taking your full course of antibiotics. So those are some things that are within your realm of control, right, that you can do to help help to prevent the spread of antibiotic resistance.
Okay, so I'm not a microbiologist at all, I'm a chemist, and there were a lot of terms when I started working with Heather that I had to learn. And one of the big differences is when you hear her talk about gram-negative and gram-positive, I was like, what does that even mean? And I liked this visual here because it really shows us that this is gram-positive and this is gram-negative, and basically lots of antibiotics will treat against gram-positive bacterial infections. Lots of antibiotics will kill those. The gram-negative bacteria are the ones that have all the tricks. They have all of the resistance kind of arsenal that they can pull, and so those are much more difficult to treat. And so really when we talk about needing new antibiotics, we're really focusing on ones that can fight back against gram-negative bacteria. So if we wanted to classify antibiotics, there's a good number of different types that we could put up. I could put lots of chemical structures up here, but I wanted to focus on the three major places where we get them and design them. And the first is, we have natural antibiotics, and these would be ones that are made by a fungus or a bacteria, and we just extract it and use it as is, right? Purify it, and then that's our drug, like penicillin, like original penicillin. Okay, so that would be completely natural. Most of our antibiotics are what we call semi-synthetic, in that we've taken them from nature, we've taken them from the bacteria produced them, and then we alter them in some way. In a chemical lab, they've worked out some kind of way to make a derivative of it and change up the structure. And a lot of that is to decrease side effects or to combat these resistant things, these tricks that the bacteria are doing. I want to show you, oh yeah, and then the last one is completely synthetic, and that would be an antibiotic that we have just made from, we don't need a bacteria or something living to make our starting material for us. We're just going to assemble them in a lab, okay? So I want to point out a couple things, and then I'm going to tell you a little bit, I'm going to explain a little bit about penicillin and how it works and how a bacteria can be resistant to penicillin, just as an example. I think it's a really interesting one. Okay, so first I want to show you just a little, for those of you that maybe haven't had chemistry or haven't had much of it, but I just want to point out the important thing to us here is this, you see this little square ring? See the square? This is a beta-lactam ring, okay? And you see it in penicillin as well. This is called a beta-lactam. And all these little places here, these are carbons. They're just not shown. Chemists understand that those are all carbons. Okay, so this is a beta-lactam ring, and I'm going to talk a little bit about that. It's an important part of penicillin. And this is cephalosporin. This is a class of antibiotics, and we are on the 5th generation of them as far as like changing them up to combat antibiotic resistance. They have altered them, chemists have altered them at this alpha 1, alpha 2, A2, A1, A3, different places for different reasons, okay? And so they're on lots of generations have come, and they keep developing new ones. So let me get back to penicillin. This beta-lactam ring, and then of course cephalosporin works very similar. What happens is penicillin enters a bacteria cell, and there's this thing called the penicillin binding protein, at PBP, so what we'll call it for short, the penicillin binding protein, and it has one job. And obviously they named it because of what penicillin does to it, but it has one job, and its job is to make the cell wall of the bacteria stronger. So what it does is it actually makes crosslinks. It builds these crosslinks in the cell wall, and it makes it stronger. So that's its one job. All right, so penicillin enters the cell, and it binds to this PBP, and then the PBP can't do its job. Okay, so it binds to it, and then after it binds, this beta-lactam ring opens, and it binds, and then it binds permanently to the to that PBP, and it can't do its job. And then what happens is the cell wall doesn't have all of those crosslinks it needs. It's weak. It has weaknesses that make it break down, and the bacteria cell dies. Okay, so in response to that, bacteria have evolved to be able to, through mutations of their DNA, they now can make, when they're resistant to penicillin, they make something called beta-lactamase. And beta-lactamase is an enzyme that when penicillin enters the cell, then beta-lactamase goes and binds to the penicillin, opens that beta-lactam ring, it opens that ring, and then penicillin can't do what it was going to do. It's not able to then bind to the PBP, and so then the PBP just keeps doing its job, and the bacteria cell is fine. And I might just point out that one of the reasons that PBP binds penicillin is because it looks to it like what it's supposed to be binding. It looks very similar structure to what it's supposed to be doing. So it thinks it's doing its job, and it's binding to something else that makes it permanently unable to do its job. Okay, so the cell then has this beta-lactamase that it makes, and so then what chemists have done is they change up like this R group here or whatever's over here, and they change that to make it where that beta-lactamase can't bind to the penicillin, and so then the penicillin can still do its job. So it's like we keep coming back with a bigger gun, It's like nature makes a way around what we designed, And then chemists have to design something else that can overcome that thing that that nature has come up with. So it's that kind of thing going. All right, so completely synthetic. I did want to put this one on here. I think it's interesting. The fluoroquinolines, they are useful. Doctors prescribe them, and I know that my mother-in-law's had some of these before, but she was pretty ill. They have now, the FDA, this is completely synthetic, but the FDA issued a strong warning last year and just said, if you have another option, don't prescribe these, because the side effects of them have turned out to be pretty severe if you do have side effects from it, and it's not worth the risk if there's another option. So we're talking nerve damage, muscle damage, tendons, and it's sometimes permanent and debilitating. So they have a new warning on this. And side effects are often a small population, but if that's you, they always want to weigh the risks of what could possibly happen with the benefit. And so now this particular class, many of them, they are only prescribing if they don't have another good alternative. Okay, this slide just shows a few that Heather's going to talk about, but these are some of our drugs of last resort. These are kind of big guns that we pull out. And I show you this to show you the complexity of these molecules too. I think even if you haven't had a lot of chemistry or been used to looking at organic structures, you can appreciate the complexity of these molecules. And I will say that it sounds simple to change up something, like we'll just change this little group over here. But it's incredibly complicated because natural products chemists have to come up with reactions that will change one part of a molecule but not mess with the rest of it. And often that includes some elaborate steps around it, or protecting this stuff over here with a reaction and then doing this other reaction and then coming back and taking these protecting groups off. And it's very complicated, and it takes many years to work out some of these, and sometimes there's just roadblocks to these syntheses. And I'm going to talk a little bit more about this in a little bit, but I think it's your turn now. We're tag teaming.
Okay, so these are our antibiotics of last resort. And so when we look at the statistics in the United States of all the different kinds of antibiotic resistance, we see all of the different kinds of strains. The strains that are resistant to these antibiotics are the ones that we're most worried about. So what are those bacteria, and what are those kinds of infections? So carbapenem-resistant enterobacteria, CRE, are a growing concern because, again, they're resistant to one of those antibiotics of last resort. So if they don't pull those antibiotics out, we hope that they have lowered amounts of resistance, but we're seeing resistance to even those last resort antibiotics. And especially the bacteria that have NDM1 or KPC2, because those are on those mobile elements, those plasmids. So when you see this particular type of strain in the news, or you read in the journals, I know you all are reading the journals, if you see these particular types of genes popping up, you'll know those are more dangerous because that gene can be shared with many different kinds of bacteria and pass through populations much faster. I've mentioned this before, but our 2-year-old from Connecticut had this MCR1 gene, which is a colistin-resistant gene. Colistin, again, is one of those big, complicated antibiotics we saw on the previous slide as an antibiotic of last resort. And then we know that there are certain classes of bacteria that we're always a little bit worried about. So Mycobacterium tuberculosis causes the disease TB, or tuberculosis. It infects 1/3 of the world's population. Mycobacterium itself is an unusual bacteria and that has an extra layer or coating on its cell wall that makes it incredibly difficult to treat. Antibiotics don't penetrate that bacteria very well, period. And so the drugs that we can use to get into this particular bacteria have to be taken for 6 to 9 months in some cases. It's a long time to be on antibiotics. So when we see bacteria like Mycobacterium that is resistant to multiple drugs, that is an incredibly growing concern. Vancomycin, resistant enterococci, and MRSA. Some of you guys have heard of MRSA. Methicillin-resistant Staphylococcus aureus used to be called methicillin-resistant. Now we tend to call it multi-resistant because these types of bacteria are often resistant to more than one class of antibiotic. And then lastly, the escape group. So this is a group of bacteria, mostly gram-negative, although we have a staph in here, an enterococcus, that we're concerned with overall. So these are the ones that are the most concerning strains of bacteria that we want to develop new antibiotics for. And I'll show you in a minute my students they work on these bacteria, safe relatives of them. We don't use the dangerous pathogens in the lab, but we call them escape relatives. And we use these classes of bacteria to screen and find those new antibiotics. So I showed you before the concern that we have with agricultural use. So of all the antibiotics that are used, only 30% are used by humans. 70% are consumed by animals in our food chain, right? In our global food web. Now you think, well, wow, that's a lot of sick animals. Actually, if you look only at the dark blue parts of the pig here and the chicken, that is the percentage of the antibiotics that are used to actually treat a sick animal. Most of the antibiotics are used in agriculture to increase growth promotion, so make the animals bigger faster, or prophylactically as prevention. So we just heard in my previous slide that humans should not take antibiotics if they are not sick with a bacteria that the antibiotic treats. Yet we use 70% of our antibiotics in agriculture improperly, right? So not to treat an active illness in the animal. Now, maybe those are different antibiotics, right? So it turns out that's not true either. So many of the antibiotics that we use in agriculture are also used in humans. So a recent study last year from China showed that over 20% of the chickens produced are resistant to colistin, that antibiotic of last resort that we were just talking about. So colistin is used in agriculture production, and yet it's our secret weapon that we keep locked away in a cabinet. So this is a growing problem that we see. Now, antibiotic resistance isn't new. It's making the news now because it's getting a little bit scarier. But from the time of discovery of penicillin here in blue in 1928 with Alexander Fleming, we've known about resistance. Even Alexander Fleming in his own notebook said, watch out, use these drugs carefully because they will become resistant. The bacteria will become resistant. And so that was first identified in 1940. And we can see that the different classes of antibiotics, tetracycline was pretty quickly found to be resistant. And this last group, the fluoroquinolones, they were actually documented resistant cases before it was really widespread, so the same year, right? So it's not a new problem, and it's not a problem that we predict is going to go away. The problem here is that in the past 30 years, there has not been a new class of antibiotics discovered, right? Now, what does that mean for us, right? So we know that we came close with our 2-year-old from Connecticut, but this is a report from January of 2017, so just a few months ago. A Nevada woman, she was 70, had traveled to India to be with family and had come back. She had had some hip and leg fractures and got an infection that was what we call pan-resistant, resistant to every known antibiotic used in humans. And she passed away from that superbug-resistant bacteria. Okay, what can be done?
Yeah.
So a lot more gloom, and you're more.
Yeah. Okay. So the Pew Charitable Trust actually published an article that's pretty interesting about sort of a scientific roadmap for antibiotic discovery. That if you're interested in reading their report, you should look it up. It's very easy to find. But basically, the ways that the pathways forward to get new antibiotics. First off, basic research. Understanding what are the bacteria doing that's making them resistant. We need more basic research funding because it's organic chemists, having better research as far as like what kind of reactions are available to them that they know they can try to make new drugs. So basic research always needs to be funded, and that's one of the things that people have a hard time funding because they want it to relate to some, like, how am I going to get my money back out of this? Or they want it to be some kind of an investment, and it's an investment in our future, really, anytime that they invest in basic research, because we're going to need it down the road. Building chemical libraries tailored for antibiotic discovery. One of the big things is sharing information, sharing options, building some kind of like systematic, storehouse for information that can be shared is going to be really important. Natural products. Now, we need to get creative, and I'm going to talk about this in just a second, but we need to start getting creative to find more new natural products. As nature evolves, as bacteria are evolving and they are getting a better arsenal, we should be able to tap into that and find better antibiotics that they are making. We need to share better. knowledge, materials, and expertise is particularly related to antibiotics. And this is much of what they talked about is what we need to happen. We need more partnerships between public and private. When I talked about during World War II, where everybody kind of got together and decided to tackle this problem together, right now, pharmaceutical companies are not interested so much in antibiotics because there's not a lot of money to be made in it. Unfortunately, if you use an antibiotic very much, it becomes not effective, right? Because resistance will be built up against it. So we're talking about needing to spend time and energy developing something that may not pay off, because to actually have a great antibiotic, it means that you hold it under lock and key and only use it when you have to. There's not money in that. So much of the work on this is being done by national labs, government-funded research in academia and those places. And but we do need partnerships where everybody's working together. Crowdsourcing is a great thing, and we're going to talk a little bit more about the Small World Initiative just briefly. We won't promise not to keep you here all night. We're getting close. So, but crowdsourcing in academia is a really good way of putting a lot of people on this project and screening lots of samples. Okay, so how many natural antibiotics do you think are found in soil? And this is your question, Joe. What do you think? Just give us a guess. 5. Okay, well, let me tell you that every antibiotic that we have talked about today, you can find that in soil. All of the bacteria, every fungi that we have talked about, we can find those in soil. And so actually the dirt is a great place to look for new antibiotics. In fact, all of these drugs of last resort are found from bacteria. They're made initially by bacteria that you can find in soil in certain parts of the world. Somewhere in the world, you can find it in the dirt. Okay, so I'm going to tell you about 2 of sort of the cutting edge, like here's the game changer kind of research things that have come out in the last few years. These are very complicated projects to pull off, but they're really cool. Okay, so this first one is more of a microbiology, and then the second one will be a chemist's approach. It was published in Nature in 2015, and anytime you have a paper in Nature or Science, those are the 2, it's a game changer. To get published in those, they're game changers. Well, this one was really cool. Okay, my favorite fun fact that I have learned in this whole thing working with Heather is that 99% of bacteria found in soil can't be grown in the traditional way in the lab. In other words, we've been going and we've been getting soil samples and we're looking for bacteria and growing them up in the lab, and we're just scratching the surface, because 99% of the bacteria that are in our samples that we collect in our yard, those of you that are in her class, 99% of those bacteria, we aren't even seeing them. Okay, so that's what's crazy. So we have all of that to tap into, potentially. Well, this group, they found Teixobactin, and it is a new class of bacteria. And it was basically what they did was they took a soil sample, they diluted it down until they got single cells. They put them in a semi-permeable membrane, which for those of you that have had chemistry, you'll know what that means. But it's basically a membrane that allows stuff to go in and out, but not the cell. That bacteria is stuck in there, almost like a little jail, a little cage. So it's like a little cage, and then they buried it in the ground. So they're in the dirt, in their little cage. So they've got the benefit of everything that's in the soil can go in and out of this cage, but the bacteria is stuck in there. So then they grew them that way. And in that, they were able to isolate this new bacteria, and so far, all of the bacteria they've tested it against, nothing is resistant. So that is a very encouraging finding, and that's why they're in Nature, because this is super cool. Okay, so, but this research, I was talking to Heather about it, and she was like, this is really hard. Like what they had to do and the amount of work they had to do to get single cells and bury them and do this work. This is very hardcore research that who knows how many and how long, and wow, this is some tough work, but it paid off big. Okay, and then the next one is the chemistry one. This one was also published in Nature. This was last year. And this is a group from Harvard. These are the macrolides. This is a class of compounds. And basically, this is erythromycin, which is in that class of compounds. They all have this big ring here. And they usually, using this as a starting point, this whole class of compounds would be made by taking this as a starting point and then changing it to make it a better drug or make it a different drug or combat some kind of resistance that has developed. Okay, but what this group did was they started with little building blocks, smaller molecules, and then they could make all kinds of versions of those, and they figured out a process for putting those pieces together to build these. So instead of making a semi-synthetic one, they built a completely synthetic one from smaller building blocks. It's a very complicated process, but the fact that they were able to build it instead of relying on nature to make their starting material allowed them to make over 300 new candidates. Where normally someone would spend years to try to make one new candidate, they were able to find a process for making 300 new candidates that now they can screen them and test them and see how they work out. So this is a pretty exciting way that chemists were able to sort of, this natural product development is, this type of organic chemistry is, these people are long suffering and they're very hardcore. It takes a long time, but they were able to really come up with a ton of compounds. So that's also a very exciting thing. Okay, we're just going to tell you about our work real quick, just a brief intro. And we'll go into detail on Wednesday next week if you come to that presentation, but go ahead.
Yeah. So in 2012, Jo Handelsman at Yale in the Center for Scientific Teaching developed a program called the Small World Initiative. And her goal was to bring authentic research into the classroom, as well as solve a global crisis of antibiotic resistance. So just a small task she was trying to accomplish. She went on to then be President Obama's advisor for antimicrobial research, one of his advisors. And what they did was, after they got the program up and running at Yale, they then expanded and trained faculty all over the country and all over the world to do this program. So you can see here a map showing all the different faculty groups all over the country. And here is our blue dot right here. This is, I started working on this and did the training and became a Small World Initiative faculty member in fall of 2014. I was then joined a few semesters later by Jamie Cunningham, also in the microbiology department. And then about the same time, or about thereabouts, we started our collaboration with Dr. Harvey. So we've been doing this for now 7 semesters in the classroom. And let me just give you a really quick overview of what that looks like. So my students, this should be familiar. Right? So students get to pick and decide what soil sample they want to bring in. We've also had environmental samples from ponds, from sand, from bee pollen, and lots of different kinds of soil samples. And they take that soil sample and they grow it on agar plates. So here, each of these little circles is a pile of bacteria we call a colony. And you can see even on this plate that these bacteria have made those picket fences, right? They've secreted that antibiotic and created a little zone around themselves. So here's a colony of bacteria and you see this little ring around it. Here's one here, and here, and here, and here. So we call these candidates, right? So the students collect these bacteria onto a collection plate or a patch plate. And students typically get between 5 and 15 different bacteria that they will find in their soil sample or their environmental sample. We then match them up with those escape relatives. So we match them up with those bacteria that we really need to find an antibiotic to. So in this bottom panel here, you can see a student's bacteria, and it's secreting an antibiotic that in this case is killing Staphylococcus. So that's one of those escape pathogens that we're worried about. Now, the rest of the semester, we do all the same things that you would typically see in a microbiology lab. We do gram staining, where they characterize their candidates through staining, biochemical and metabolic testing, and we send the samples off for DNA sequencing. So they get to identify their bacteria using a genetic approach. At that point, we have a pretty good collection of viable candidates that I then hand off to Melanie's class.
So one thing that makes ours unique here is that, as far as we know, we're the only partnership between microbiology and chemistry. And you actually approached me really early on, and I started working with honor students, but it was hard to accomplish a whole lot. It's just been much more productive to have a whole class of students participating. So we're a little unique in that, and part of that comes with that is we're making it up as we go, because we did not have a lot of colleagues to ask, hey, what are you doing to go, what's your next step? So we started it off just sort of making up as we go, and then we did find one group at Yale that had partnered with a chemist for a while, and what they were doing was pretty much what we had decided to do. So that was like, all right, we felt really good about ourselves because we had, separately come to the same conclusions that that's how we should approach this. So right now, my Chem 140 lab, and I have one student here tonight that's in that class. What we're doing is we basically take plates of the bacteria that we're interested in looking at, and we're using a certain protocol to extract from that and try to isolate whatever it is they're making that's killing other bacteria. We want to try to isolate that antibiotic. And so then we try to separate it using a technique that we would do in this lab anyway, which is TLC. It's a classic organic chemistry thing. Chromatography, it's where you separate parts of a mixture. And then we put those in plates. We send them back to Heather. She's not done. I keep her busy. So she send it back to her, and she pours agar on them with bacteria embedded in it. And basically we see, did we isolate something that's inhibiting growth of bacteria? And then we've gone on to do things like try separating them more and isolating those components. This is the part we're kind of working out the details of right now. But we have had some interesting results, some promising ones, as far as like we're able to isolate something that does inhibit growth. So we feel like we're on the right track. And we're sort of able to do this with a whole class of students. It's a good way to sort of screen all the candidates. And then we can spend more focused time on the ones that look more promising with the different procedures that we're using right now. But we're also always kind of improving our procedures and our approach and trying something new. And we've tried a number of things in just the few semesters that I've been doing with this class. We keep making it better, and it's going better every time. The best part is that all of our students get to participate in authentic research. And so we're really interested in the outcome, but we're also interested in the process and the opportunity that our students have. And it's a pretty unique one in that a lot of people don't get that kind of experience, especially not in your first year of a science class. So that's a pretty cool thing. Okay, so we've done poster sessions every semester with our students where they present their research, and they can put this on their resume, present it at an actual thing because it's totally legit. People go around and grill them and everything. No, not really. No, just kidding. No, actually, people are very interested in what you're doing, so they'll come around and they'll ask you what you did. I'm not just scare them. Okay, very good. So, anyway,
it's like in 2 weeks.
No, it's very exciting. It's very fun to show off what you've done. Okay, and so everyone, science is always a community thing. And so it's a very cool thing to be a part of something like this. So we've done poster sessions every time, and we have had so far over 400 student researchers that have participated here at Johnson County Community College. We have sequenced 189 different bacteria. We have presented 191 posters and done chemical analysis on 46 different samples so far. So it's been a really cool thing, and it's something where we've been able to include a lot of people, and they get to be a part of it. So it's been a great opportunity. Okay, and I think we're done. So if you have questions for us, I know we kept you a long time, but there's 2 of us, so we talk a lot. But if you have questions, we'd love to hear them right now. So. Yes, Joe.
The proliferation of the antibiotics in livestock, what effect is that having? Because it's pretty pronounced. And is there any research going on here in the food corridor on that?
There's not. There's a lot of work way higher than my pay grade in the government trying to understand the impact of that and how do you control that. So again, it's a bottom dollar issue, right? So they want to make the biggest chicken the fastest and the cheapest because we all want our chicken nuggets to be $1, right? And so it becomes a problem with trying to impose regulation or impose sanctions or even taxes on those antibiotics. And it's not just the United States, it's a global problem. So 20% of chicken in China is resistant to colistin, has bacteria that are resistant to colistin.
Yeah, and you know, we don't have a culture that likes regulation on things, especially if it's going to cost. And so that is the problem. It's like, who's going to tell me I can't do this? And like you said, you know, when it's an international issue, those are even harder to get everybody on board. We're so global that we're so interconnected that what other countries do and the things that they cause to happen, we're all affected by it. Everyone's affected, including we affect other people, they affect us.
Matthew?
Piggybacking off what you were just saying and what Joe had brought up as well, are there any international organizations working to remediate livestock antibiotic usage or the overusage on the chicken farms especially?
There's a number of nonprofit groups that are trying to impact change and bring awareness, and the World Health Organization has it on their radar, but unfortunately it's very difficult to get many countries to sign on to regulations like that. Yeah.
When the body's fighting off any kind of infection and it creates antibodies, do those antibodies not help at all to fight off the infection? And it can, like what happens with the killer T cells in antibodies means trying to fight the infection?
Absolutely, yeah. So the time before antibiotics, right, we had to rely on our natural defenses. And so antibodies are proteins that our body makes as a natural defense to the pathogens that we're infected with. So they certainly do work in any infection process, hopefully if you're healthy. But antibiotics are just a way to avoid that infection. Antibodies take about 2 weeks to develop fully, the highest number we get. And so we don't want to be sick that long. So we use these drugs to help reduce the length of the infection and get healthier faster.
It seems a lot of the research is in kind of wet labs. Is there any computer simulation that can be done that could possibly go a lot faster?
Yeah, so this, was it this fall?
Yeah.
Ooh, it seems so long ago.
No, wait. What semester is this? This is spring. It was in service this semester.
It was in service,
January.
okay. So in January, I brought in,
Sorry.
a lot has happened this semester. In January, I brought in some training from a group called Genome Solver. And one of the things they do is trying to bring authentic research into dry labs or lecture settings. And they use a bioinformatics approach. So they taught us some tools to search databases of peptides and to look at modeling and trying to predict genetic elements that might be important and doing that dry lab part of it, doing a computer bioinformatics approach. But so far, we just had the training in January, apparently, and we haven't had a full implementation of that yet. Barry?
30 years ago, what was the trigger to stop the development historically? What factors were involved?
Yeah, the pharmaceutical companies, there's a great documentary called Hunting the Nightmare Bacteria, and it's on PBS. And they interviewed different pharmaceutical companies to find out exactly why did they drop out of that market. And they'll tell you it's a portfolio decision, right? So there are drugs like cholesterol-lowering medications and ADHD medications and antidepressants that people take every day for the rest of their life. Those are way more valuable to that for-profit pharmaceutical company than these antibiotics. And even vaccines, which is another way that we can combat infectious disease, everybody, hopefully, right, gets vaccinated. And so that's a lot more profit for that pharmaceutical company than a drug that, again, you don't really want to use for a long period of time.
It's almost like a cultural shift in our culture towards money driving everything instead of, you know, I mean, science has always been, you know, sometimes you're just curious and you just want to make things better and you're not thinking about, you know, making a buck. So, other questions? Well, thank you guys so much.
so much.
You've been a wonderful audience. We appreciate it.